Here's a very cool bacteria, the Thermus aquaticus. Thermus aquaticus is a type of extremophile and thermophile bacteria, which means it enjoys very, very high temperatures. It was originally discovered in a hot spring in Yellowstone National Park, and originally it was thought that nothing could survive in these pools and springs because of the extreme temperatures, but this bacteria and a couple others were found to actually love living there. Originally, when this bacteria was first discovered, the scientists that found it were just kind of like, neat, and then left it alone for a few years because they didn't think very much of it. However, it turned out to be actually a very, very important bacteria. It has a specific enzyme called Taq DNA polymerase, which is used in one of the most important, cellular and molecular biology techniques ever. Polymerase chain reaction, or PCR. I have talked about PCR before. I've talked about doing it in my different lab classes I've taken. I've also talked about Kary Mullis, the guy who's first invented it before. But this is the bacteria that is responsible for it. It was the first one that the Taq polymerase was ever discovered in, and it's very important because it's super, super stable at super, super high temperatures. And by isolating and using this enzyme, it was found that you could use it in a laboratory setting to amplify and make new DNA, which is very important in so many different settings and it has so many different uses. And it is all thanks to this little bacteria that was found to be living in one of the most extreme conditions on the earth. Use this sound, all of the proceeds from the artist go to Palestine, Congo, and Sudan.